Okay, so welcome to the fifth video in our course here on F1 flow cytometry analysis. So here we're going to look at the practical elements. So we're going to look at the practical element mainly for slow flow cytometry. But we're also going to touch on what we're going to do for the parts on G-plate and bioassays as well. So what we're going to do is divide our experimental flow cytometry analysis into different stages. Firstly, accessing the raw data and the FCS files as a flow set together, a bunch of different files. We're going to compensate and look at data transformation as well. We're going to look at gating and separation of the cell populations. And then we're going to look at summarization and how to visualize that data set effectively using a range of approaches. An example of packages that can do this is Cytometry. So that's an example of that looks at trajectory for changing patterns over time. It looks at visualizing flow and mass cytometry data. And it can do a range of different features like dimensionality reduction and so on. What we're mainly looking at is it for a kind of illustration of the steps that we're looking at. So think about it, we're using typically a bunch of different FCS files and going through this process of compensation, gating, normalization, concatenation. You can see that written as OR code down here. So we have a function that takes in board an argument of the raw data and creates an object out of that called site. And then we can use our code to process that. And then we do things like trajectory analysis, where you might run some clustering approaches or build a tree, for example, for further analysis. And that's shown over here that we might want to look at, say, the dimensionality reduction to see if we can reduce the dimensions that are present. And then look at our cell types to discover their relationships. And in this diagram, what we've got is different cell populations shown by 10 different clusters here. So what it will do is try to route these different clusters as shown here for 1 and 2. And look across the different graph to identify the shortest paths between different clusters. And what it'll try to do is track paths from its route 1 and 2 to the furthest point in this cluster. So if we know the cell type of cell group 10 here, I know 1 and 2, we can identify what are the intermediate states of 4, 7, and 9, because they're obviously important for the transition between 1 and 10. Whereas for 5 and 8, they're not as important because they're on the periphery of that network. So it's really representing the cell types as a kind of a tree-like structure. So for the first step, what we're doing here is just taking on the different flow cytometry files, performing standard steps to assess them together as an object, which is just a type of representation of the flow cytometry data that makes it amenable to different functions and packages to evaluate it further. So what we can see next is this process of creating a minimum spanning tree from our cells based on clustering and dimensionality reduction, and using many dimensions of coordinates to create this kind of tree-like shape. This is run by using different functions that operate on our object. We'll be using a slightly longer version of this for our lab. And what Cytometry is doing is trying to run pseudotime analysis, which identifies each of the cells present as a point connected by edges. It looks at the k-nearest neighbor algorithm to kind of map out the routes between the different cells. We might have certain cells that are known, like 1 and 2 here. And they might look at the proximity compared to cell type 10 and see what paths 1 and 2 would wander through. So you can see here that 1 and 2 would wander through mainly 4, 7, and 9, but also could go through 3, 4, 7, and 9 in the case of cell type 1 as well. So what it looks for is the shortest walks from the root to the leaves, and these represent the intermediate states. We have other applications as well. So for looking at homeopathic differentiation hierarchy, we can look at these in a tree-like pattern, where we have a known representation of what to expect in advance. And this is in human bone marrow, and the cell types are multipotent progenitors, myeloid progenitors, and other lymphoid progenitors as well. So what we can do then as well is plot a tree of the different cell populations that are based on manual information. So that's kind of represented here. Then here where we've taken our data of the different cells, transformed it in an appropriate manner, looks at the dimension reduction approaches, created a minimum spanning tree to represent them, and then with no markers, labeled the different cell types here, as shown by this kind of legend, where we have ones that are clearly identified as different, whereas there's some down here in the middle which are more difficult to distinguish. And therefore, we can also identify what markers are most associated with the different cell types and see how they differ as we go along. There's a range of different tools that we'll be using, and these are listed here also at the start, and these are mainly in bioconductor, where there's different packages that we use, one of which is Flowcore, another one is GG Cyto, another one is I took from is RG Lab 2. There's other packages that exist out there that we're not using. There's many different ones. These are for GG Plate mainly, called Plate Tools, Plater, and PhenoScreen. But these are harder to use. In our GG Plate lab, what we're going to do is make plots of culture plates and microplates using appropriate plate layouts, ID 96, 48, 384 ones, like you might have here, where you have the measurements inside each individual well that's of interest. And we're going to use some code from this location here as well. Similarly, in bioassays, we're also going to look at different plate formats from 6 to 384. And we're going to see how we can use CSV data, import that, and send that to our plate to represent it in different ways, depending on what's of interest. So that's what's coming up in our labs. Please see the demonstration videos for more details on what we're actually doing in those different labs, where we'll talk about flow cytometry analysis and GG plate and the bioassays as well. So please look at those videos in the lab 1 and 2 folders.